thank you so much for the invitation to talk about my research today. And it's a fantastic opportunity to interact with you all online. My name is Taekwon Ko. I'm working at Michigan State University and Plant Research Laboratory. The work that I'm talking about today is one of my postdoc projects, but I'm actually working on more exciting new project. So if you're interested, please send me email related to anything of science or careers and anything I can help or collaborate. And I apologize, my voice a little bit low because I had cold and still have it. And let's get started. Can we change this? Okay. Right. So I thought it'd be nice if I can introduce what I'm doing in Michigan here. My research is heavily associated gene regulations. So gene regulation is a very complex process and is primary response to development, growth, and stress responses, metabolism, for many biological processes. But it's mainly achieved by interplays between cis and trans-acting elements. So cis-acting elements is known as cis-regulatory elements called CRE, is short DNA sequence and gene promoters. And the trans-acting elements is protein that transcribe some arrays like transcription factors and bind the CRE, the sequence-specific matters. And this interplay between the cis and trans-acting elements mainly triggered by internal, external stimuli. And the second value of my research is genomics. Genomics is a unique way to get the collective characterization and quantification of genes. It cares about data generation at large scale. It aims to assess biological questions with genome-wide perspective. And while genomics cares about a lot of data generation, it is also important to interpret the data properly. So third value is network biology. So the main principle of network biology is molecular components like genes and protein interact with each other to create biological networks, so which allows complex organisms like plants to coordinate biological response properly. It aims to explore how the relationship between these individual molecular components give rise to complex emergent properties. So my research used the power of genomics to attain the systems level understanding of gene regulations in plants and revolutionizing the crop improvement. I use not only computational tools, but also a lot of experimental tools. So that's what I'm doing on my research. Just summarize one slide. And let's go back to the project. So my postdoc research, I focus on understanding how genes respond in environmental stresses. But specifically, I was interested in how the stress signal gets sensed within cell and delivered to the nucleus. So I thought ER stress is kind of best example of how this process is going on. So let me introduce what ER stress is. So ER, the endoplasmic reticulum, is cellular organelle. It's very important for many molecular and cellular metabolic events. So the main job is to fold proteins properly and dispatch them right place within cell. In normal condition, perfectly good, because there's balance between protein folding demand and protein folding capacity in ER. When plants exposed to stress like drought and heat or the pathogen attack, the protein folding demand increase substantially. When it arises beyond the ER capacity, the lethal condition known as ER stress can develop. So you can imagine like traffic in rush hours, a lot of proteins jam the same highway compared to the just normal conditions. The response to ER stress highly conserved not only plants, but also across eukaryotes. So it involves in diabetes, cancers, Alzheimer's disease, and many other human diseases. So better understand the ER stress not only important for agronomic losses, but also the human disease drug development. So to maintain ER homeostasis, the plant initiate conserved set of signaling pathways known as unfolded protein response called UPR. So UPR is very unique. So there's two important things. It has kind of two strategies, adaptive strategy and apoptosis pathways. So in the beginning, the plans, the UPR, so mainly control this ER homeostasis. How? Increasing the number of the proteins, folders like chaperone, or reduce number of proteins translating. When ER stress persistent and it's not resolved, the cell recognize, okay, this is not gonna work. So it changes the pathway to kind of program cell death. So it basically just kill themselves. It's called apoptosis pathways. But this transition is controlled by the dynamic gene expression changes. And not only is transitions from the beginning of the actuation of the UPR, the transition and termination of this UPR all controlled by dynamic gene expression changes that associate cellular metabolic events during ordinary ER stress. So in plants, ER stress mainly controlled by two branches. One is involved in membrane-associated transfer factor, BG28. The other involves in membrane-associated dual functioning protein kinase ribonuclease, IRE1, and its splashing target, BG60. So upon ER stress, BG28 moving to relocate to Golgi, where it is progressively processed by post-translation cleavage that it releases the nucleus. And upon ER stress, the IRE1 is being activated, and it does the unconventional splicing of the mRNA encoding PG16 mRNA, PG16 encoding mRNA. They create a frame shift. Now this mRNA now encodes transcriptional nuclear targeting signal with this frame shift. Then PG60 moving to the nucleus. So PG20, PG60, they are heterodimerize, homodimerize, and jointly regulate their target genes. Okay, this is kind of basic schemes. But there are a lot of knowledge gap how 2060 regulate, the dynamically regulate, what regulate is 2060 kind of signal. So we, in the lab, we are very interested in identifying kind of unknown regulator in the UPR. So one of the approaches we took is four genetic approaches. So by mutagenize UPR mutants using with the mutagens called EMS. So EMS is chemical mutagen that mainly induce the GC80 transition randomly in plant genomes. It's widely used in plant genetics. And basically we mutagenize UPR mutants and the screen the mutant that has a kind of suppressed phenotype of this lethality and isolate the mutants and map the mutation in the genome. And once identify the mutation, where this mutation is, then characterize the gene that has a mutation. That's the main strategy. So I'm showing some set of example here. So this plate showing half is DMS, which is the solvent, the more control. The other half, the growth media containing tunicamycin. Tunicamycin is bestly known ER stress inducer. So DMS basically is solvent of this tunicamycin. First row showing the types, Columbia is wild type. So wild type has little problem is ER stress condition. Of course, a little sicker than more control. We can see it can grow. So basically it can overcome with all these UPR components. It's okay. When you remove IRE1-AB, I just show you IRE1-AB. IRE1 is proteins can sense the stress signal, can splice PG60. So if I remove the IRE1A, the proteins, plants is really sick. You can see it can barely grow. This is suppressor of this IRE1-AB. So we mutagenize these IRE1-AB mutants and see they partially suppress this lethality. So this IRE1-AB and PG60 triple mutants, high order mutants, plants really sick. When the suppressor is underneath here, you can partially suppress these lethal phenotypes. P2060, two transvector gene, they are knocked out and see really sick. So we have these two independent suppressor mutant 2060 can partially suppress the lethality. So as you can see, this is a very simple technique and powerful to identify the hidden regulators in the complex regulatory system. So you can imagine like you're trying to find the needle in the haystack. The small tools like magnet is really helpful, can expedite the process. The simple is very useful. So the EMS screening is kind of play that kind of role in plant genetics. So, so we screen a lot of mutants, and specifically I was interested in the suppressors that can suppress the lethality of IRE1-ABs with heritable and recessive phenotype. That's what happened here. The Colombia grow, wild type grow like this, and IRE1-AB mutant is very sick, dry weight, fresh weight, primary root, that's all gross phenotype. This ISM93, this mutant can partially suppress this lethality. So we name it ISM93 because it's called IRE1-AB is a suppression mutant. And one thing I want to mention is that this mutant, IRE1-AB, this mutant is naturally a shown root phenotype compared to Colombia. Not even in non-stress condition, DMSO conditions. This shown root phenotype was not affected this suppressor mutant, which means ISM93 alleles specifically linked the stress responses rather than the physiological development. So you can see Colombia's grow about 90% Colombia compared to DMS more control. IRE1-AB is about only 20%, and ISM93 about 40-60%. So partially suppressed this lethality. That's what I isolate this mutant. And this mutant, we isolated and it's mapped where the mutation is. So common technique we use is called bulk segregant analysis. These techniques only effective the mutant is recessive homozygous. Dominant mutant cannot be isolated with this technique because the principle of this bulk segregant analysis is segregations. So as you know, dominant mutant cannot be segregated. So to perform the bulk segregant analysis, I create two populations, a susceptible, resistant population. Let me explain everything here. So I have ISM93 original mutants, M3, then backcross IRE1-AB. So all these green triangle, there's SNPs. So EMS create boom, like hundreds, thousands of the mutations. But through this mutant isolation, I was able to identify this mutant controlled by single mutations because I identified Mendelian genetics 3-to-1 ratio. Otherwise, it cannot be a 3-to-1 ratios. So I knew that one mutation is responsible for this phenotype, but I also understand there are a lot of 100,000 of mutations not linked to the phenotype. So green is SNPs not linked to the phenotypes. And the red star is basically causal mutations here. And I backcross in the F1, there's a heterozygous. And F2, they let them select. So F2 generation, there's a kind of two populations. Either they have resistant to ER stress or susceptible, like IRE1-AB, or like partial suppressed IRE1-AB resistant. They found this ratio 3-to-1 because these Mendelian segregations. These populations that have all these red star homozygous, but these guys may have wild types, no red star, or red star, or heterozygous. Then I whole genome sequence this resistant pool, susceptible pool, and IRE1-AB, ISM93. Basically, I'm trying to identify the earlier the highly mutants have high frequency and resistant phenotype like red star, but susceptible line may be low frequency. And the 100% frequency ISM93, the genotypes, but 0% frequency of IRE1-AB. That's what I wanted to identify. So as I told you before, the mutation, the EMS caused hundreds of large mutation. So that's what happened here. It's just simple map. And there's always single, it is circle that indicating the mutations, and different color indicate different types of mutation. So mutation can occur in intergenic regions, or intron, or exon 3.5 UTR. But I was specifically interested in the mutation can cause amino acid sequence changes called non-synonymous mutations. So I was able to identify here this chromosome 2 has a non-synonymous mutations with high frequency about 75%. So let's talk about in detail. So chromosome 2, the mutation around here, it turns out its mutation locates first exon of gene encoding PIR1. Okay. So here this genome browser showing. So this reference sequence is guanine G, but the mutations has changed to A. So the ISM93, FPC1, F2, these two populations, resistant populations, basically suppress IRE1-AB lethal phenotypes, have 74% of this A, but this frequencies dramatically reduce the population of susceptible population 38%. And IRE1-AB has 0%. And ISM93 original mutant line have 100%. They're indicating this mutation might be responsible for the phenotype we observed ISM93. Then what is PIR1? The PIR1 is the plant-specific link type ubiquitin E3 ligase. It has established E3 ligase activity, so indicating these mutations and involved in this ubiquitin ligation process. And one of the beauty of this four genetic approach is that you never know what you're gonna get. We never worked on E3 ligase stuff before. Now we have kind of opening door for our new reactions. So this is what I found, this mapping, but I have to validate this mutation really responsible to the phenotype. So I did complementation analysis. So I introduced the genomic fragments, the PIR1, promoters, and all this entire genomic fragment, introduced this fragment into ISM93. It turns out ISM93 is complementation, independent complementation line, have the partial suppression of these suppressive phenotypes. So it's not going to be 100% suppression for this nature of this plant transformation, but we genetically validate, okay, so this mutation first axon PIR1 has fully responsible for the phenotype we observed ISM93. All right, so PIR1 is key component ubiquitin proteasome system, or UPS, so which is suppressed by IRE1 now. So I hypothesized that UPS could be activated in the stress, to participate in the cell fate determination coordinated by IRE1-AB. So I tried to measure the global level of the ubiquitination, Colombia and IRE1-ABW mutant and ER stress condition. So here, this Colombia, this ubiquitination of this ubiquitin conjugates, ER stress, the tunicamycin added plus minus mean DMSO. You can see even as early as 1 hour, the ubiquitination conjugate level is highly increased, even early hours, 1 hour, very early stage Colombia. But in IRE1-AB mutant, this induction of this ubiquitin conjugations dramatically increase in the absence of IRE1-AB. Then we also check, let me see, the phenotypes of the plants. So we treat the IRE1-AB and 2060, the UPR mutant, which MG132, which is proteasome inhibitors. Interesting is this proteasome inhibitor treatment can revert the lethal phenotypes of IRE1-AB, not only IRE1-AB, but also PG28 as well. So they're indicating UPS is not only activating ER stress, but also necessary for the execution of ER stress-induced cell death due to insufficient UPR. And now to better understand how the PIR1 that works in the UPR. So now we identify PIR1 as new regulators. Now we have vision to identify the molecular mechanism, how PIR works in the UPR. So we perform RNA sequencing analysis because I told you before, dynamic gene expression changes one of primary response to the ER stress. So we did RNA-seq. So this volcano plot showing Colombia first, genes that are downregulated, genes that are upregulated. You can see a lot of genes upregulated compared to downregulated genes. In the absence of PIR1, the gene upregulation significantly, globally is induced in the PIR1 mutant, like this. So global gene induction level was enhanced. So I performed clustering analysis then, all the genes differentially regulated and just clustered them, cluster based on expression patterns. I found 4 clusters, and here cluster 1 and 2 specifically have group of genes which gene inductions was enhanced in the absence of PIR1. So without PIR1, these genes is highly, with PIR1 genes induced, but in the absence of PIR1, it's more induced. That's what I'm talking about. So a lot of UPR biomarkers in P3, ERGJ3, A, and B, they are all sharpened on 4 days, and PG6 that I told you before. And those genes that belong to the cluster 1 and 2, but IPG28, IRE1A, those genes already known to be less responsive to transcriptional level ER stress condition. This gene is not effective. So I hypothesized that the PIR1 could mediate the stability of transcription factors that activate the expression of those genes. So to test these ideas, I performed a de novo multiple analysis. Basically, I grabbed the 1 kb promoters of the gene that belong to each clusters and find any cis regulatory elements in each of these gene promoters. They found out ACGT, which is G-box, is significantly enriched promoters of gene cluster 1 or cluster 2. Okay. So then to identify what could bind this G-box, which is significantly enriched promoter of gene belong to cluster 1 and 2, which means those genes are highly induced in the absence of PIR1. I look at the DAPSIC database. DAPSIC is a technique that identifies protein transfectal DNA interactions in vitro in Arabidopsis. So GPAX is a core sequence known to be bound by PGIP and BHLH transfer factors. In this database, there are 30 and 28 PGIP BHLH transfer factors respectively. I scan all the transfer factor binding patterns. My parameter was whatever interesting to bind to specifically B3, BGIP6, which they belong to cluster 1 or 2, but not BGIP28, which is belong to cluster 4 or 3. So then I found out few among them is ABI5. ABI5C specifically bind to B3 promoters here, or BGIP60, but not BGIP28. And their core binding sequence is ACGT. And a few years ago, I also published another paper showing in which I performed enhanced East-1 hybrid. So East-1 hybrid, as you know, protein DNA interaction screening. The enhanced East-1 hybrid is a technique that's semi-automatic. So they have 2,000 transfectal libraries that screen basically all the DNA fragments that I'm interested in. So we identify 100 of protein DNA interactions. Then I look at this, my database, and found that ABI5 is only transfer factors that bind to P3 promoters or PG60 promoters, but not PG20 promoters. So we have kind of strong kind of candidate to can furthermore the investigative the ABI5. So we performed the Western blot first in PIR mutants. It turns out ABI5, the protein levels is significantly actually more stable compared in PIR1 mutants compared to Colombian. So ABI5 proteins are more stable in PIR1 than Colombia 0, the wild types. So at this point, we got this kind of connection between PIR1 ABI5 because we have protein DNA, we have this DNA binding domain, DNA binding data, and also we have this protein level change data, but there's no still knowledge gap between ABI5 and the UPR. To identify this knowledge gap of ABI5 and UPR, the functional relationship, I perform chromatin precipitation assay called chip assay. Now, to identify binding of the ABI5 in P3 and PGIP60 promoters and DMSO and ER stress conditions. Basically here, actin7 is whatever sequence enrichment here, they are basically the background level. So you can ignore whatever sequence enrichment of so below here. And ABI5 indeed bind P3, but the binding was not changes. DMSO and tunicamation. So ABI5 bind P3 promoters. This binding intensity was not changed. And I found ABI5 protein binding to PGIP60 promoters. And interestingly, this binding intensity has been significantly increased in stress conditions. So it seems like ABI5 bind both P3 and PGIP60, which is consistent we found in vitro, the DAPSIC data file, or in East-1 hybrid. But in vivo condition, we found out ABI5 more strongly bound PGIP60 promoter, but no change in P3 promoters. But binding doesn't necessarily mean transcriptional activity. So we have to check whether ABI5 binding is actually doing something on gene regulations. So as we perform transient gene expression analysis in tobacco. So ABI5 effector alone, so there's two kind of construct reporters P3 or PG60 promoters driving luciferase gene. Effector is kind of overexpress the lot of transfer factors or UPR regulators. So first, ABI5 effector alone did not increase the transcription level of P3, which is consistent. We found the chip assay data. So ABI5 doesn't necessarily change the P3 expression level. But the additional ABI5 effector cell significantly increased the activity of luciferase driven by P60. So it seems that ABI5 function is a kind of activators UPR by directly inducing the expression of P60 but not P3, but P60 induce P3. So kind of signal cascade. ABI5 induce P60 expression, which then the increase expression of P3. So we check the molecular level, then I want to check the transcriptional level, the genetics. The absence of ABI5 knockout mutant, P3 and P60 expression actually substantially decrease in ER stress conditions. So it's still induced. They are still induced, but the absence of ABI5 is not as much as wild type. In ABI5 overexpression 9, when I overexpress ABI5, the P3 and P60 expression is substantially increased. So they kind of explain ABI5 function as activators in the UPR. The phenotypic level, and I check the phenotypes of ABI5-8, which is ABI5 knockout mutant. Knockout mutant hypersensitive to ER stress compared to the Columbia in the vary of dosage of ER stress condition, different concentration of tunicamycins. That means that if you remove ABI5, plant sicker than Columbia. So here I'm showing kind of summarize what's going on here. So UPS is main function to try for cell death by reducing the abundance pro-survival factors such as ABI5, likely others. And as you know, E3 ligase, they have multiple targets. So we are looking at basically a lot of different targets. ABI5 could be one of them, but it could be indirect regulation. But we have checked whether PIR1 is ubiquitinase or ABI5. We could not find direct ubiquitination of ABI5 by PIR1. So we believe there's kind of an indirect regulation PIR1 ABI5. And on the control of IRE1-AD, which also coordinates the expression of UPR target gene as cell survival strategy. Right. So this is very nice, intra-lab collaboration project in the brain disease lab. And I'd like to thank all the co-authors for this important piece of data, piece of papers. And I was fortunate to have a small team with exceptional technician and the graduate. And that's it. And I'm happy to take any questions.